The word quality has been part of the Upjohn motto for 100 years. Ensuring that quality is the job of the Control Division.
Welcome to the Control Division. We wish we can open all of our laboratories for you to see in person. Because that's not possible, we've produced this videotape to show you some of them. I hope that viewing it will give you an understanding of the vital role our employees play in ensuring the quality of Upjohn's products.
In Control, our mission is to ensure that quality is designed into our products during development and that the products we manufacture maintain their quality throughout the production process and in the marketplace. In our business, we can only be sure we have a quality product when we know all we can about even the smallest component. We must subject everything that goes into a product, as well as the product itself, to painstaking tests. That requires sophisticated equipment and dedicated, highly trained people. We're about to visit some of them. The first area we'll visit is the Control Development Group, which is responsible for setting quality standards for new Upjohn products. Laboratory scale synthesis of bulk drug is used for studies of safety and efficacy, formulation development, analytical specification development, and synthetic process evaluation. Later in the pilot plant, techniques may differ, but the goal is still to efficiently produce analytically pure material. Chemists develop specific and accurate chromatographic control test assay procedures for bulk drugs and pharmaceutical dosage forms. This helps us obtain the information necessary for filing a new drug application or NDA. Laboratory personnel interact with a computer in the characterization of pharmaceutical products. Data is collected by computers and later recalled by the chemist for careful analysis. Peaks on the computer's screen correspond to amounts of specific compounds. Another important technique in product characterization is the use of high vacuum system technology. Knowing how a compound degrades when exposed to heat or oxygen helps to characterize it. The high vacuum system is used to evacuate air from the sample and replace it with oxygen, which speeds up the oxidative degradation process. The goal of the mass spectrometry lab is to employ current instrumental techniques and to develop new techniques of analysis for solving chemical structure problems. Each potential new product must be characterized for compounds which are either impurities or stress degradation products. The spectroscopists aid in the identification of these compounds with the help of instrumentation, which uses a combination of electric and magnetic fields to determine the molecular weight of an unknown compound. The Nuclear Magnetic Resonance Laboratory has two spectrometers to aid the scientists in determining the structures of various drug candidates and their byproducts. Placing certain types of nuclei in a magnetic field and measuring the radio frequency energy absorbed results in a spectrum of resonances. The spectrum provides information from which the compound structure can be deduced. Customer complaints, deviations in raw materials and packaging components, and problems in manufacturing processes frequently require the identification of small particles and other trace substances. Chemists in the Trace Analysis Laboratory use sophisticated microscopic techniques to analyze particles as small as a few microns. This work requires a wide range of micro-manipulation techniques and instruments, including polarized light, stereo, scanning electron, and infrared microscopes. The protein sequenator is a high-pressure liquid chromatograph that helps us to identify amino acids at particular positions in the molecular chain. Determining the sequence of amino acids is an important part of the quality control of protein drug candidates. The laser system records the color of the fluorescent light emitted by the sample and times how long it takes for the emitted light to appear once the light pulse has been directed onto the sample. Our principal use of the information from time-resolved fluorescence spectroscopy is to study the static and dynamic molecular structure of proteins through the measurement of fluorescence spectra and lifetimes. Results from scientific investigations are summarized, interpreted, and documented in technical reports written by Control's research and development staff. The reports, along with seminars and symposia, provide scientific communication among scientists in the Upjohn technical community. Test assay procedures developed by Control scientists are issued as drafts in the Control technical report. Their evaluation culminates in an official approved test assay procedure maintained in a VAX computer database. Having procedures available in a computer database allows Control personnel to call up a copy of the current test assay procedure at any of many remote terminals throughout the Control Division, including some of our overseas locations. This collection of tests and assays forms the basis of Control's portion of a registration for a new product with the FDA and international regulatory agencies. After quality standards are established, the Control Assay Service Laboratories provide accurate analytical data for the acceptance of materials, the release of finished products, and the evaluation of product and material stability. The analytical data is then provided to the domestic and international quality assurance organizations. Chromatography separates components of a mixture. This method is used to determine the purity of raw materials and the potency of a given drug in a product. A small portion of a liquid sample preparation containing the drug is bottled and placed in the autosampler of the chromatograph. The autosampler injects a portion of the liquid onto a column where the drug components are separated, identified, and quantitated. As part of Control's program to improve productivity, a GC-LC robot automatically prepares samples for chromatographic determinations. The operator places single tablets into sample preparation vials. Then the robot adds a small amount of water to each vial, causing the tablets to break apart. A solvent is added to each vial to remove the drug components of interest from the tablets. A small portion of the liquid containing the drug components is placed in chromatographic bottles, which the operator transfers to a chromatograph for analysis. The FAST LC is a completely automated chromatographic system which prepares and analyzes the vitamins of A, D, and E in a tablet. The operator puts the tablets in cups where they are pulverized and the vitamins to be analyzed are removed into a liquid. A small portion of the solvent is injected onto a column where the vitamins are separated and quantitated. Infrared spectroscopy is used to confirm the identity of many incoming raw materials and Upjohn products at various stages of manufacturing. For most identifications, the sample is prepared, smeared onto a plate, then placed into a beam of infrared radiation. The instrument then measures how much infrared energy is absorbed by the sample and at what wavelength absorption occurs. A plot of this information is called the infrared absorption spectrum. For most organic compounds, this spectrum is unique and can be used for identification by comparing the sample spectrum to that of a reference material. In the X-ray laboratory, the analysts perform two distinct types of X-ray spectroscopy, fluorescence and diffraction. In X-ray fluorescence, a sample is bombarded with X-rays, and the resulting X-rays emitted from the sample are monitored. The emitted rays, which are characteristic of the elemental composition of the sample, are used to identify trace impurities in the sample. In X-ray diffraction, an X-ray beam is directed at a crystalline sample, which scatters the beam in a well-defined pattern. This pattern is used as a fingerprint of the sample, which can then be compared to known reference patterns and matched for identification of the crystalline structure. In the microturb plate testing laboratories, we determine antibiotic and vitamin potencies. Antibiotic assays are based on the premise that an antibiotic, when added to a particular bacterial growth system, will inhibit growth. The potency can be determined by comparing the amount of inhibition to a standard curve of known concentrations. Vitamin assays are based on the principle that they will enhance bacterial growth. In addition to laboratory testing of final products, the quality assurance units monitor the production process throughout manufacturing to ensure that our quality standards are maintained. Raw material inspectors retrieve sampling plans for each raw material from the computer. They inspect the material containers for obvious defects and verify receipt of the correct material. Then, following the sampling instruction, a sample is obtained for laboratory testing. Many sterile products are formulated in two-compartment vials for improved stability and convenience of use. Control inspectors routinely audit samples of these freeze-dried products for appearance, force of activation, and reconstitution time. Results of these tests are recorded on lot records and become part of the batch review and release. Tablet products are also routinely monitored during the manufacturing process. Among the improved checks are tablet weight, hardness, and friability. Control inspectors perform periodic fill weight checks during the packaging operation to monitor the accuracy of the filling equipment. Pre-weighed empty bottles are placed on the line and sent through the product filler. These bottles are removed after the labeling operation and taken to an inspection station for weighing. A personal computer interfaced to an electronic balance automatically records the fill weight of each bottle. The computer then graphically displays these fill averages so that line operators can monitor the efficiency of the filling equipment. In addition to fill checks, control inspectors also audit the packaging process to assure that the correct labels and package inserts are used. Newer methods of in-process testing are also being developed. Near-infrared reflectance analysis is a technique that uses the diffuse reflectance of a sample. Using NIRA as an analytical method enables us to simultaneously analyze water and lincomycin in soybean meal feed. In this project, the goal was to come up with a single method capable of rapidly doing all of the product release tests, which consisted of water content by loss on drying, potency, and identification of lincomycin by gas chromatography. Because no sample preparation is required in the NIRA method, control now has a nearly instantaneous measure of the quality of lincomix. Results of the in-process testing by QA inspectors and laboratory assay results are electronically summarized for review and disposition by the responsible QA person. Electronic release of the batch transfers it into the finished goods inventory for distribution to customers. Samples of each Upjohn product are stored and then tested at various intervals for up to 6 years. Members of the Stability Administration Unit evaluate data to determine the product's shelf life. Shelf life is the length of time that we assure that the product will meet all Upjohn quality standards. The Word Processing Center supports the control organization by providing word processing, document duplicating and distribution, mail delivery, and support services to all control units. We employ well-trained personnel and contemporary information processing technology, including word processors and personal computer hardware and software. The technicians in the Sample Coordination Center are responsible for receiving, processing, and distributing all types of materials and products from throughout the Upjohn Company to the control laboratories for testing. Stability samples from the units are also submitted to the Development Center, already matched with the appropriate records for the control stability system. Retention of samples from production lots for possible FDA use is required by current good manufacturing practices. Control Computer Operations provides computer support for a wide range of activities in control. These services are provided on a total of 9 super mini computers and a rapidly growing number of microcomputer systems. Facilities maintained for communicating with these systems include a local area network, links to the administrative computer center, and a telecommunications connection that permits access to and from our overseas subsidiaries. User support and programming services are provided to control units requiring integrated computer systems to assist control staff in meeting their goals. Upjohn sales abroad amount to over 40% of its worldwide total. We manufacture our products at the international locations shown on the map. The Control Division is directly responsible for the assurance that consistent standards of high quality are applied wherever the Upjohn name appears. More than 400 quality control personnel are employed at these international locations. Likewise, development of products overseas is becoming increasingly important, with major facilities located in Crawley, England, and Takasaki, Japan. The Japanese research facility currently employs about 70 researchers and support staff. In 1985, the Upjohn Company announced significant expansion plans in Japan, where a new laboratory will be constructed to accommodate up to 400 researchers in lead finding and new product development. The Control Division, as a worldwide leader in the design of product quality standards throughout all phases of product development, will ultimately employ more than 40 people at the new site in Japan. Whether a product is developed and produced in Kalamazoo, in Japan, or in other subsidiaries, product quality is the principal objective of control employees. The product specifications developed during the research phases will ensure that Upjohn products remain the highest quality the industry has to offer.